So today I'm going to talk to you about ADC characterization on a single platform. This is with an instrument that we're calling Q Exactve BioPharma. So this is kind of a rather large jump from my proteomics days in the Yates lab, but you know, if I didn't do that, I don't think I'd be doing this. So thank you, John. Anyway, so ADC characterization is quite complex. You know, so any time that we have a biologic or a therapeutic protein, basically this is a complex mixture. So comprehensive characterization of trying to account for these possible modifications, oxidation, deamidation, scrambling of disulfide bonds, possible sequence variants, you know, this is something that requires multiple approaches, and it's the cumulative effect of these multiple approaches that allow us to understand these very complex molecules. And as Michael pointed out this morning, some of these things, carbohydrate structures, for instance, you know, this is something that can have a rather large effect on the efficacy or the function of a drug that we're trying to make. We're relying on cells, which are biological factories, and sometimes we can control them to some extent, and sometimes we can't control every aspect of these biological factories. So we use these assays, molecular weight, drug to antibody ratio, chain pairing, subunit analysis. We look at these molecules from multiple angles to understand what the grand picture is. So in this case, characterization, I think a lot of folks that do characterization work, the main workflows that are being employed are intact, subunit, and peptide level analysis. So here, with these icons that I'll use throughout my talk here, with the intact antibody, the subunit either reduced or processed by the Ides enzyme and then reduced, or the peptide mapping approach, you know, putting all of these data together is what really allows us to understand these incredibly complex molecules. So the Q Exactve BioPharma is something that this wasn't our first attempt at getting a mass spectrometer right that's perfect for characterization. What happened? Well, in 2011, we launched this Q Exactve mass spec, this platform that really, right off the shelf, is fantastic for peptide mapping. Thermo mass spectrometers have been really well known for being the leaders in proteomics, and that extends directly to peptide mapping. Most of the technology is identical. So in this case, it's also lent itself well to middle and top down. This is looking at medium to smaller proteins where we're actually doing MSMS and we're understanding these incredibly complex fragment patterns. So the Q Exactve can do denatured intact analysis. But really, in 2013, at the end of 2013, we launched this Exactve Plus EMR, which stands for Extended Mass Range. And this mass spectrometer, we quickly found out, was really the very best. Not the best for Thermo, but the best in the industry for intact protein analysis. This was capable of not only denatured approaches, but also native conditions, so doing native mass spectrometry. So as we learned this, as the application side of things caught up pretty tremendously, we found out how to automate some of these things. So whether that's in fusion or now we're doing chromatography for native mass spec, we found out that in certain conditions, you know, we could tune the optics and get an even more optimized result. So this Exactve Plus EMR represented basically a quantum leap in intact protein analysis. Again, not just for Thermo, but across the industry. So what we did was we took really the feature set which was appropriate for antibodies and ADCs, and this represents a great deal of the market for biologics. So basically we adapted this functionality of the EMR, and we created a new mode which is capable of native intact analysis, denatured intact analysis, and we've wrapped this into a mass spectrometer called the Q Exactve BioPharma. So to take a step back, we're extremely proud of this mass spec because in the greater scheme of things, it represents one continuous platform, including the Vanquish UHPLC system, the Q Exactve, and this comes in two flavors at the moment, the Q Exactve Plus or the Q Exactve HF BioPharma. The BioPharma is an option that you can add on to either of these mass spectrometers. And then finally, the BioPharma Finder data analysis software. So all of these three in line represents one platform, extremely powerful, which is ideally suited for characterization. So the Q Exactve BioPharma has pre-optimized workflows. I mentioned a moment ago that we implemented a mode that's specific for intact analysis. So that's called high mass range mode. When we analyze these antibodies in either denaturing or especially in native conditions, the spectra, I'll show you examples of this, the spectra show up at the very, at much higher reaches of the spectrum. So higher M over Z ranges. So this high mass range is very helpful for that. Protein mode is really the go-to mode for using the highest resolution powers of the Orbitrap, and using this on small to medium-sized proteins or subunits, and getting this, it says here, extreme resolving power of the Orbitrap. That might sound extreme, but this is true. This is the mode. It's ideally suited, the gas pressures in the HCD cell, and you basically turn this on and it's already optimized for subunit analysis. That extends actually as well to top and middle down. So I'll show some examples later in the talk about how you can go from a subunit analysis to actually generating MS-MS information. So protein mode, and then finally standard mode. This is, we're calling it standard mode. To be honest, when it was implemented, there was just it. So peptide mapping, though, is what these instruments were initially very good at. They're still very good at them. So by changing the modes and not changing the hardware, we can change the way that the mass spec operates with the same tried and true hardware that has been good at protein mode, subunit, top, middle, down, and peptide mapping. And now we have high mass range mode. Okay, so a few notes here on the BioPharma Finder data software. So this is ideally suited for all kinds of intact analysis. And I'll get into some of these details here, but essentially for the different flavors of intact analysis, you might be using different deconvolution algorithms. We've got those. So for peptide mapping, BioPharma Finder is also ideally suited. So this is software which is incredibly powerful and can do the discovery level identification of various PTMs, and it does this in a peptide mapping fashion. This is peptide mapping BioPharma software. This isn't proteomic software force fit to the biopharma world, but it's actually specifically designed for peptide mapping in biopharma. So there's also additional features which I won't really go into, but there's a ton of other stuff in this software. One grand package, it's actually very powerful. So I spoke about the different kinds of intact analysis that you might want to do. Basically, we have roughly two flavors. I'm calling this, in this case, intact analysis or intact MAB, and then a MAB subunit. So in this case, for an intact antibody, actually what we would do, we would turn on the high mass range mode, and with this, we would be using a lower resolving power in the Orbitrap, and we would generate average mass measurements, or average mass data, and we would measure an average mass of each of the species in the mixture. So this is actually not an isotopic profile, but rather this is actually a glycoform profile, and underneath each of these peaks would theoretically be isotopes, but this is something at 150 kilodaltons, a very large molecule. So when we generate these kind of data, we actually use the RESPECT deconvolution algorithm. So this is sort of represents one workflow here. On the right-hand side, the subunit analysis or subunit workflow. In this case, we would use protein mode. We would generate data which is isotopically resolved. This is really specifically for small to medium-sized proteins. So in full disclosure, there is a mass limitation on getting these kind of data. And as you jump over that mass limitation, this would be different for a plus, a QE plus, or a QEHF. As you sort of surpass that mass size, then you really want to be using this high mass range mode. So if we have questions on that later, I can answer after the talk or in person. But the point is that, there we go, high resolving power, isotopically resolved, and we're using the extract deconvolution algorithm. So this is a completely different workflow, distinct from the intact workflow. And we utilize them both, and the BioPharma Finder software is able to cater to both of these. So when you actually turn on the software, the way it looks is you're presented with these experiment types, which would be peptide mapping or intact protein analysis. And both of these experiment types refer back to the protein sequence manager. So this is where all of your sequencers are stored. And if it's peptide mapping, this is where we generate our sequence map. Or if it's intact, this is where we do our annotation to figure out what each of these peaks represent. Okay, so this software supports all Orbitrap mass spectrometers. Actually, it supports ion trap mass spectrometers as well, but the difference in resolution will obviously give you different kinds of data. Ion traps being low resolving instruments and therefore not capable all the time of achieving the same kinds of results that you might get on an Orbitrap. Now again, I mentioned before there's two deconvolution algorithms, respect and extract. This would be for the intact and the subunit analysis, and I'll get into great detail here in a minute. But something that's really important, this is a major, we call it a differentiator. This is a very, very unique facet of this software, particularly for Thermo, something called sliding window deconvolution. So sliding window deconvolution is totally unique, and well, it is deconvolution that's finally appropriate for separations-based mass spectrometry. So I think we all really just want to put the sample right in there, but the complexity of the sample oftentimes means that we need to separate this out from other forms. And getting that separation, I think we can all appreciate that the data become even better. But once we start separating things, the data are different. So what do I mean by that? Well, as we begin to separate, this is an example of an ADC. This would be the MAB, the antibody with 0 drugs on it. And then this would be the antibody with 2 drugs and 4 drugs. And what you see is that as we add more drugs, this red trace in the middle represents the extracted ion chromatogram of these different species. You see they're not eluting all at the same time. So in the conventional deconvolution approach, for those who haven't done it, what happens is we actually, we're going to make an average across the peak. And then we generate a spectrum, and then we deconvolve that spectrum, we figure out what's there. But if you've done this before, you'll know that exactly where you make this, this average, I think I'm out of laser juice, exactly where you make this average is very important, and it can shift the information, it can shift the readout. In fact, that user bias has a tremendous weight on the result. So what we've done is we've figured out a scheme. This sliding window deconvolution, what it does is it performs a series of spectral averaging and then deconvolves each of these average spectra and then stitches these together. So we start at the beginning of a separations experiment. This could be liquid chromatography, this could be CE. Either way, we start at the beginning and then we move through the chromatography, or the separations, and then we end at what we determine to be the end. So after doing this, you can see that these would be the individual spectra. Now I did this manually because what happens is when you do this automatically, it represents a very familiar output, but these are actually now peak areas, deconvolved species. So this is in the mass domain, not M over Z domain, and it stitches these together to create these peak areas represented by intensities, and this is automatically integrated. You ask, what is the difference in the result? Well, the difference is twofold. The biggest difference is that we have designed a method where we are not intervening as an individual analyst. And in the end, you, me, everyone in this room could press start, and we would get exactly the same result. Once you've set up the method, it processes exactly. So there's a lot that can be done here. So batch automation is a big deal. I missed my cue in the last slide, but basically all biologics are benefited. Simple MABs, complex ADCs. The fact that you can do this exactly the same way and you can move through large batches, this is a really big deal. The other big deal is that you're actually going to get the right answer. And so this talk is on ADCs, and ADCs represent something unique in the intact protein analysis world because there's a type of information that we can only access at the intact level. And this would be the drug-to-antibody ratio. And I'll talk a little bit more about this in a few slides. But what you can see is that when we compare the sliding window to the conventional deconvolution method, you can see that it's very difficult to get the right answer because of this phenomenon of things eluting at different times with the different elution profiles. So up top, we have the sliding window analysis, and you can see that we're getting sensitive data all the way out to 9 drugs attached to this antibody. When we look on the bottom, we can see that especially at these higher drug loads, that we're not getting the same answer. So what's the right answer? Well, the sliding window is the right answer because what's happening is these higher drug loads tend to elute later. They tend to elute later in the chromatography. So if we're making an average across the peak here, we're not capturing any of these things. So we're biasing the result against the higher drug load. So this is the correct result. It's also the easy way to do it too. It's also the consistent result. So as we get into this high mass range mode, I mentioned earlier that we can do intact analysis now with either denaturing conditions or with native conditions. The next question you might have is, why are you giving me choices? What's the best one? Well, you know, mass spectrometry and characterization is not necessarily a one-size-fits-all scenario. We have different biologics that we're working on, and they represent different challenges. So this is essentially a fork in the road where you can generate the same answer, but the denaturing approach versus the native approach, these offer different advantages depending on the actual complexity or lack thereof, really the challenge at hand for that particular protein that we're analyzing, or other biologic for that matter. So in this case, I'm showing denaturing MS data. The data shown here are Trastuzumab, and in this case we have a reverse phase column, and so using the organic and the formic acid in this case, these are both properties in solution phase that will denature our protein. So with reverse phase chromatography, we're generating now a mass spectrum which is sort of in a lower end of this M/Z range with an apex charge state of 49 plus. And you can see if we zoom in on that one charge state, we have a profile that looks like this, pretty iconic actually for Trastuzumab. And then alternatively, we can generate the same data, really the same, if we look at this in this native data, and we look at this very specific charge state, 25, or the other charge states for that matter, the profile is exactly the same. So we're getting the same result. We actually do this in an automated fashion now. So this is native mass spec. This is not infusion. This is not nanospray. This is incredibly easy to do if you've ever used an autosampler, if you've ever used an LC or a mass spec, a column, then I think you'd be capable of doing this. So we're using size exclusion. We're using volatile solvents here, or rather volatile mobile phase, no solvent. So this is 50 millimolar ammonium acetate, excuse me. And we generate a peak. Actually, if you've ever done native mass spectrometry before, it's an incredibly painful, we'll call it art. So if you've done this before, then you know that you're actually manually filling these glass capillaries, these emitters that you have to position perfectly in front of the mass spectrometer to actually get data. But before you even do that, you have to go offline. You have to buffer exchange this into these volatile salt buffers, which can accommodate this physiological or physiologically like structure, but also then removes all these mass spec unfriendly salts. So in this case, we're using size exclusion to do this automatically. It actually is desalting on the column. So we'll come back to this in a minute. What's the difference though? Why would you use one versus the other? Well, denaturing mass spectrometry is really, it's compatible with separations that are very familiar, reverse phase separation. It's also very sensitive. So the organic, the formic acid, forcibly charging these proteins, it lends itself to very sensitive detection. And this is really kind of the tried and true method for intact analysis so far. So this is a new world, and we found out that native mass spectrometry is very good for these heterogeneous mixtures, ADCs, glycoproteins, pegylation. And this is going to give us the most confident analysis. So I'll show some slides on this soon. And actually, the chromatography we would use for this is size exclusion. This allows online desalting. Okay, so for denaturing analysis, I mentioned that this was more sensitive. In this case, I'm showing, again, Trastuzumab. This is online using a Q Exactve Plus, of course, with HMR mode, high mass range mode. And this is using our MabPack RP column. And in doing this, we can run this fantastic range, demonstrating sensitivity all the way down to the 0.1 nanogram or 100 picogram level. I'm showing a deconvolution at 500 picograms, and we're getting very nice mass accuracies and the same masses as we're getting at a much higher level here at 100 nanograms. So, you know, this is a valuable approach for a great range of sensitivities. When we get into ADCs, now this is another ball of wax altogether. So ADC construction makes a complex mixture that is an antibody. It makes it even more complex. It's nearly over the top. And we found that the denaturing approach really wasn't suitable for this, to get the right answer consistently. So what we've got here, this is a cartoon depicting an antibody which has its glycoforms, and as I just showed a moment ago, it's not one glycoform, rather it's a distribution of possible glycoforms that can be on this antibody. And so we create some heterogeneity with that. There's also possible variants, sequence clipping variants, you know, maybe lower abundance, but nonetheless they're there. So this diversity is compounded by making a conjugation. So adding these linker drugs, in this case, the line and the star, and adding several of them. So this is a chemistry experiment. We're going to create a distribution of forms on top of the mixture that's already there. And I'm also depicting this little line here. This is the linker without the drug on it. So this is a byproduct, which is not intended, but as you do organic chemistry on this biological mixture, sometimes the organic chemistry isn't perfect either. So this contributes then to the heterogeneity that is introduced at the conjugation step. And that's depicted here. In this case, this would be a two-step conjugation, adding the linker and then adding the drug. Sometimes the drug doesn't go on the linker, or maybe we have a bad linker in the mix. A lot of things can happen. So to demonstrate my point between denaturing and native conditions, when we run this in denaturing conditions, we get a familiar mass range, but these peaks, it's just a pile of peaks on top of peaks. And so at this 2,000 to approximately 4,000 range, we're getting really just so much information. These charge states, the neighboring charge states, are overlapping and interfering with the neighboring charge states. So in this case, we have a plus 51, and this plus 51 has basically information overlapping from the plus 53, the plus 52, the plus 50, the plus 49. You know, these charge states, there's a great deal of overlap here. And as we move to the native conditions, again, this was done online with our size exclusion, and what we find is that overlap is greatly reduced, minimized. And as we continue this, if we push this even more, then there's absolutely no overlap at all. And what this does for the mass spectrometer is it makes it incredibly, well, this is really for the analyst. It makes it incredibly easy to get a confident result. So in this workflow, we generate a mass spectrum, and we can zoom in and see one of those charge states. So all of this information comes from a single charge state cluster at the plus 25. So there's literally hundreds of things in this tube, in this sample mixture. So as we look at this spectrum, if it's our job to do this analysis, then we have to do the deconvolution, but at the end of the day, we have to say, does this deconvolution result look like this? That's the challenge of somebody who does intact protein analysis. And by doing this, by preventing the situation where we have this much overlap and complexity, and then when we move to something where there is little to no overlap, and we compare that to the deconvolution result, well, the deconvolution software, we can allow it to achieve a better result, a closer to perfect result, but the analyst also can say, well, gosh, that looks pretty similar. Look at that. Yep, I like it. It's good. Let's go. So the answer that we get allows us to really pick into the data and then make additional conclusions. Not that, sure, that's sort of where my protein should be, but you know, we're just going to forget about the other peaks. Well, in this case, if we zoom in on the deconvolution result, so this is mass, and we're looking at the, in this case, this is the 1, 2, 3, 4, the D4 cluster. we're going to have information here from the glycoforms. We're going to have information from the different amounts of drug. And we're also, as we see, these are not just random small peaks. We can depict the free linkers here as well. So as you go through and label, and I wasn't extremely comprehensive. There's actually additional peaks here that we could label. This is a fantastic way of looking at something that we're calling now true intact. So we haven't manipulated our sample. We haven't taken this ADC and realizing that we've put all these drugs on it, but it also has all these glycoforms in this mixture. Now, if we deglycosylate, we can simplify it tremendously. What I'm trying to say here is that with an Orbitrap, we have additional power that we can look at this kind of heterogeneity. We actually, in this case, did not manipulate this sample. So all of this information is here with less work, less sample prep. So the BioPharma Finder software was actually very helpful in this case because from the different amounts of drug, in this case, we don't expect that our glycoform distribution is going to change. Now, that may not be the case for every single biologic, but in this case, that's what's happening, or at least that's what we believe. So based on that assumption, I actually did this DAR calculation. Again, I haven't deglycosylated the sample, but I've done now no sample prep. I've introduced it straight. We desalted online. And now I have all these glycoforms. And I can actually make use of this information. I can do this drug to antibody ratio calculation automatically in the software by highlighting the different glycoforms that I'd like to look at. So in this case, these are the 4 most abundant glycoforms for this Trastuzumab Emtansine, this ADC. And at every single drug amount, it's calculating the weighted average of how much drug there is. And we did this 4 times, and as you can see, the answer is actually quite consistent. So the top 4 average calculated automatically by our software, 3.65 is something that we have a lot of faith in. Okay, so I'm going to turn the page a little bit to subunit analysis. So again, same mass spectrometer. All of this was done using a Q Exactve plus BioPharma mass spectrometer. So in subunit analysis, the subunit analysis workflow, we're going to use protein mode. And in this case, we're going to look at an LCMS analysis, the Ides-digested, and then reduced Trastuzumab. So we have here, right, Ides and then reduction, which creates our 3 pieces, the light chain, and then the 2 pieces of the heavy chain, the FD and the FC. And as you can see here, we're actually using the same column, the MabPack RP, and we separate quite nicely these 3 different subunits. And from that, then we can use our sliding window deconvolution to generate one single result. So, you know, I guess the conventional way would lead you to 3 different reports. You do this once, you average here, and then you export your result, and that's your result for the FC, and then you do it again for the LC, and then you do it again for the FD. Or if you're very meticulous, you might do a front, middle, back analysis of all 3, and now you have 9 reports. So we can do this in 1 report. It's actually quite easy. And of course, we're getting isotopic resolution on these subunits, so the mass accuracy is incredible. The extract deconvolution algorithm, which we're going to use for the subunits, this is something that actually includes information for the mass accuracy of every single isotope. And again, we're doing this online. This was a Q Exactve Plus with a resolution setting of 140,000. We can actually then pick these peaks. In this case, this was a targeted analysis. We can pick this for HCD fragmentation. So you can do this at 1 fragmentation energy or multiple, and you can put this information together. And then you can generate basically this fragmentation pattern in the MS-MS spectrum. So here we have the M over Z domain, the mass spectrum, and then we deconvolute this using extract. Again, this is MS-MS information we've deconvolved to get a mass domain fragment pattern. So when we were doing this initially, what we've done is we've been using this ProCyte Lite. This is freeware. It comes out of the Kelleher lab at Northwestern University. And this has been incredibly powerful for allowing us to basically compare these mass domain, these fragment maps, and mapping them to the actual sequences of the subunits. So this has been a cooperation of using this freeware with our BioPharma Finder software. And as we move forward, by the end of this year, this is actually going to be a new feature in the new software moving forward. So this is going to make an actual, for the first time, workflow for middle down analysis for biopharma. And we think this is going to be incredibly powerful. This will have some features that weren't really possible with doing this in the manual approach, so it's very exciting. I'm going to turn the page again to peptide mapping. This is in standard mode. Of course, actually, when you operate the instrument, there's no standard mode. It's just the way that the mass spec is as you turn it on. So for those who are using these Q Exactve instruments, maybe that's something I should point out. But peptide mapping, this has really been our strong suit at Thermo for quite a while. From the days of proteomics, carrying it forward, continuing to lead the way in peptide mapping. So with our BioPharma Finder software, I think it's fair to say that these peptide maps contain a great deal of information. And so piecing this back together with respect to what we think the protein is and where these various sites might be, this is aided tremendously in our BioPharma Finder software by these very cool graphical sort of depictions. So we've got a lot going on here. I'll start at the top, and I'll talk about this again coming up. Basically, these little tiny lines here, this is my attempt to fit a great deal of information on one slide. These are all amino acid letters. So these bars here are actually peptides, which are spanning the sequence. And as you can see, they're different colors. So the different colors are, it's a heat-coded intensity depiction. So we're actually looking at this incredible dynamic range, but we're also looking at this incredible breadth of coverage in sequence. So this is a heat-coded sequence coverage diagram. This is one of the very unique features with the software. One of the things you see at first, the very first thing, you get your result back, and actually we're looking now at these various species that we would expect to be in our sample, and then if they're not there, we're actually also looking at unidentified. So in this case, I did 3 analyses of, this was approximately 60 minutes times 3, and all of these data were analyzed at once, and the sequence coverage, of course, is 100% for both. Now, there were some identified peaks, so when we look at the total peak area, we see 60, approximately 30, and then a little unidentified. Now, when we come down to the chromatogram, we can see that that identified stuff is entirely here at the beginning. This is where our small molecules are eluting right away. They sort of blow through the column. They're not peptides, so they're not going to match to peptide spectra. So here they're getting gray bars. We're actually color coding the chromatographic peaks, and this is information you get right as you turn on the software when you get your result. So we've also got the heavy chain and the light chain, and these color-coded bars are accounting for every single thing that the software has identified or not identified, so essentially everything in your sample. I'm pointing out one peak here. This is a glycosylated peptide. So this is where our glycan is going to sit on the consensus sequence. And BioPharma Finder also allows us to automatically identify these glycosylated peptides and determine exactly what kind of glycosylation is on this peptide. So in this case, Michael mentioned earlier that we're using this kinetic model. And I'll just say one thing about the kinetic model. It's essentially this incredible way of understanding how different combinations of amino acids will fragment and produce different abundances of these fragments. So it takes into account a great deal of academic literature, mobile proton. This is sophisticated stuff. And what you can see is this software is predicting a spectrum, and the acquired spectrum is nearly identical. So again, this is the predicted spectrum for an unmodified peptide at this site. We're getting this great sequence coverage. And then in the acquired spectrum, it's labeling all those peaks. It's actually going one step further, and it's using this heat-coded sequence map idea even at the MS2 level. So this is the kind of detail that we're going into for our MS2 detections. And it's taking it then a step even further. And it can do this now for glycosylated peptides and understand exactly what the glycosylation was. It can do this from, of course, the MS1 mass, the parent mass, but it can also do this from the MS2. So here we have a predicted spectrum. which is what we expect that this peptide will look like when it's fragmented by HCD fragmentation at this HCD fragmentation energy. So it's taking all of this into account. It works for ETD as well. So we have a predicted spectrum. It compares very nicely to the acquired spectrum. And what we find here is that the software can annotate a great deal of information even about the glycosylation itself. So we have a glycan-specific fragment. We have 2 glycopeptide-specific fragments. And then, of course, we have all the peptide-specific fragments. So there's a great deal of information here. At the end, this delivers a chart which is talking about, in a site-specific manner, all of the peptides which correspond to a modification at that site. And this is really, for a peptide mapping approach, the kind of information that you want, where we want to eventually be able to say, you know, what's the abundance of this form, this specific kind of modification, given the fact that there's this diversity of other forms as well. So we can do that, and this is an example of our ADC, where I ran this 3 times, and we can see that even, you know, I cut this off at the top 15, but you can see the answer from these technical replicates was actually very, very good. So this is below the 1% level, and the answer is essentially spot on. This is for an mannose 4 form, so it's not the most abundant one in the Trastuzumab Emtansine sample. Okay, so I'm getting close to wrapping up. So this is an ADC talk, so I'm going to talk a little bit about what happens when you're looking at ADC conjugated peptides. So the software does an incredible job, again, with fragmentation, annotation, understanding what this fragment pattern is, and then mapping that back onto a sequence, and it does this with great precision. Now, when we conjugate these peptides, we're introducing an unknown. So the software at this point, full disclosure, is not able to say, you know, I understand every single molecular component, atomic component in this molecular formula of your linker drug. But we can be good analysts, and we can take it one step further in the software. So in this case, what we've done is we've allowed the software to identify all the peptide-related peaks. And we know, you know, looking at the raw data, and from the literature, so we found that there are several peaks which could be, they're signature peaks for either the glycopeptide, in this case, or, the conjugated drug linker peptide fragments, or in this case, just signature ions that would be specific to the linker drug itself. So this 547 is pretty well known for this DM1 linker drug. So in this case, we're manually checking all the data that we're getting, which would correspond to an ADC conjugated peptide. So 547 is one clue. Another clue is that actually a very unique aspect about this linker drug is the fact that it has a stereocenter. And what this does in the chromatography, we find that it consistently makes a doublet. Now that spacing of the doublet might change, but there's always a doublet. So what we've done then is we've asked the software to look for these drugs. We put in the mass, and then I was able to manually confirm this. We found 30 of these peptides in this case, again with the 547 and then the doublet. And we can map this back onto our heat-coded sequence map. And again, these number bars here, these will change depending on the data. But you know, we've always got these 5 grades of intensity. So in this case, I've just manually depicted where all of the lysines are at. And then I use this orange or yellow color to depict where the ones that I found are at. So in characterization, whether it's process or whether it's very upstream, trying to understand if we are essentially breaking our antibody by adding drugs aggressively in a place that it shouldn't be. This is a very easy way to understand that kind of work. So I'm all done, but I just want to review. The Q Exactve mass spectrometer now has a biopharma option. So Q Exactve BioPharma is something that has 3 modes, and this is perfectly suited for the top 3 workflows for characterization. And importantly, intact antibody and ADC analysis is really now, you know, we've moved, we're in the future. Intact analysis is best done on an Orbitrap. And if you're going to do intact analysis, you want to use high mass range mode. So protein mode is great for subunit analysis as well as top and middle down. This requires very high resolution. And it requires operation of the instrument in a completely different manner than what you'd have for the high mass range mode. And it's yet again a different manner than what you would have for standard mode for peptide mapping. So final summary, the biopharma option is something which allows you to do complete protein characterization with one mass spec. The biopharma finder software is really exactly the kind of software you would want to handle the data coming from a mass spec, which would give you either intact, subunit, or peptide mapping. Characterization, of course, is very complex, which requires that you have a mass spec and a software that can handle those kind of data. And the structural fragility and complexity of ADCs is really something that we think is best tackled by native MS, and that's accessible using the Q Exactve BioPharma. So just thank you very much for letting me talk with you today, and happy to take questions now, or you can approach me individually later if you like.